Hello, Biology 230. This is Dr. Gerald D. Griffin, and I am coming to present to you today on the topics of mitosis, meiosis, and the regulation of cell division. This presentation will cover chapters 12.3, which is the last portion of chapter 12 in your book, as well as the entirety of chapter 13 in your biology Campbell textbook. How did your parents pass on information to you? Why do you have your father's eyes? Why do you have your mother's nose? Etc., etc. This all represents the phenomenon of heredity, which means that there's actually information passed from the parent to the subsequent generation. This is the flow of genetic information. Heritable information is passed from generation to generation through genes. Genes are located on chromosomes, and they are stretches of DNA that are actually going to encode for parts of or whole um, parts of the information that allows for the expression of proteins, which is the active players in our cells that we've talked about earlier. Parents will send their genes to their offspring by specialized cells called gametes, and it is the fusion of these gametes, which is reproduction, that will allow for the creation of a new individual, the daughter or the son, and allow for the passing along of genes into a viable individual. Gametes, compared to somatic cells, are produced and primarily housed in the gonads, our reproductive organs. Somatic cells can basically be expressed everywhere. Most likely not the gonads, but we do have somatic cells, of course, in the gonads as well. Gametes are haploid, which means they only have one set of chromosomes. You can do just HA as half, because the number of chromosomes in your gametes will be half of those that are in your somatic cells, which are diploid. Diploid, Di meaning two, just means that the somatic cells will have two sets of chromosomes. So in humans, our diploid number is 46 in our somatic cells, where our haploid number is 23, which will be in our gametes, okay? Sperm for men and the eggs or the ova for women. To transmit genes to future generation is the main function of gametes, whereas somatic cells can have multiple functions depending on what organ or tissue types they are comprising. Gametes are reproduced and divide and go through division through meiosis, whereas somatic cells will create new somatic cells through mitosis. Now we've talked a little bit about reproduction, the fusion of different gametes, but that is representing one type of reproduction called sexual reproduction. Sexual reproduction involves two different individuals, whereas asexual reproduction is merely making a clone of yourself and passing that along to the next generation. Here's a lecture assignment. I'm going to center it so you can really see it, okay? I want you to list two advantages of asexual reproduction. You need to also list two advantages of sexual reproduction, and then list one disadvantage of each of these two. So total, you're going to be listing 5 things. You need to email that to your professor. That would be me, Dr. Griffin, ggriffin@mitu.tuskegee.edu, or Dr. Bradford, whose email is cbradford@mitu.tuskegee.edu. Okay, you need to do that before this module is due. Compare and contrast asexual versus sexual reproduction. We're allowed to go through reproduction because of chromosomes. Chromosomes are the long stretches that are distinct from one another that contain our DNA, which is tightly wound on proteins on the chromosomes. Here you see two different chromosomes that are comprised of two sister chromatids. Okay, here's one sister chromatid, which is bound by its sister chromatid by a centromere. The centromere is the portion of the chromosome that holds the two sister chromatids together. Sister chromatids will be identical to each other. They are copies of each other. However, homologous chromosomes will have the same karyotype staining pattern. And right here is just an example of a karyotype. Here is the human chromosome set, all 23 chromosomes. And it is of a male because there's a Y chromosome here. And the chromosomes actually stain in banding patterns called karyotypes. And homologous chromosomes will have the same karyotype banding staining pattern. They will have the same genes. They will have the same centromere position. However, they could possibly have different versions of the same genes, which are known as alleles. One important note I want to make is that the centromere holds together the two sister chromatids by a protein called cohesin, and that's in your book, Cohesin, cohesin. Just to drive the point home about haploid and diploid and homologous chromosomes, you see here that the red represents the maternal set of chromosomes, and we have 3 chromosomes coming from the mother, and we have 3 chromosomes coming from the father, the paternal set of chromosomes. Sister chromatids of one duplicated chromosome. Again, just driving to the point that these sister chromatids are bound together by the centromere, and they are exactly alike. They are a result of replication, of that DNA replicating. Now, here are 2 non-sister chromatids, this from this blue here, and this from this red here. They are not exactly alike, okay, but they are homologous, which means they have the same genes, they have the same pattern of genes. They just have different versions of the genes, okay? And since they're not connected together and they're not exactly alike, they're called non-sister chromatids. This cell we would consider would be diploid, which means it has 1, 2, 3, 4, 5, 6 pairs of chromosomes. It has a full set of chromosomes, 1 from the mother, 1 from the father, 1 from each parent. If you have 1 from each parent, you are diploid. Your cell is diploid. Okay? The haploid number in this case would be N equals 3. That is your haploid, Haploid number. Throughout our life cycle, we go through different stages. Our cells go through different stages of being haploid or diploid. Gametes, like I said, are haploid. When fertilization takes place, you have one haploid cell join another haploid cell through fertilization. What do you get? A zygote, which is a result of fertilization of 2 gametes coming together and fusing to make a diploid cell. This diploid cell is going to divide through development and mitosis, and eventually you would rise back to make some more gametes as this individual matures in his gonads, and you're going to make more haploid gametes, and this individual will meet someone else, they will fertilize, and keep the cycle continuing. Now we're going to focus now on meiosis, which is the major topic of this presentation, but we will at the end compare it to mitosis. Meiosis is the process of how gametes are produced. Okay, it's how the sperm cells are produced. It's how the ova, the egg cells, are produced. Okay, The parent cell for these gametes are actually diploid. And we'll learn more about that when we talk about reproduction in depth. But the parent cells for sperm, the parent cells for the eggs are actually diploid cells. They have a 2n number of chromosomes. How they are formed, how these gametes are formed, is through the process of meiosis. The major result of meiosis, and this is something that you definitely want to know, is that one parent cell, which is diploid, will give rise to a set of 4 haploid and unique daughter cells. Now let's go through and see how this happens. You start with this diploid cell here that has duplicated and has one pair of homologous chromosomes, one chromosome from its father, one chromosome from its mother. These chromosomes are homologous. Okay, this is a diploid cell with duplicated chromosomes. It has 2 chromosomes. Okay, that's the diploid number. That's the 2n number. Through meiosis 1, because meiosis is divided into 2 major parts, these homologous chromosomes are going to separate. Okay, these are homologous chromosome pair 1, homologous chromosome pair 2. They're homologous to each other. One pair is going to go to one cell, the other pair is going to go to a second cell. Okay, these cells now are haploid. How many chromosomes do they have? That's right, 1. They have 1 chromosome. Here they had 2 chromosomes made up of 2 sister chromatids each. So you had 4 sister chromatids in one cell. Now you have 2 sister chromatids in each cell. You have 1 chromosome in each cell. Now through meiosis 2, these cells are going to further divide. And now you have 4 different haploid cells. They have 1 chromosome each. This chromosome now is merely 1 chromatid. Why is that? Because these chromatids have not duplicated yet. Remember, diploid cell with duplicated chromosomes. These cells with the chromosomes have not replicated yet, and we only have 1 chromatid, which is going to be the 1 chromosome in these gametes. Please make sure you understand this, okay? Go through it, read the book, let's ask questions, graduate for sure, office hours, email me, etc. Here are the major phases of meiosis 1. Again, meiosis is broken down into 2 parts. The first is prophase 1, okay? We have our nuclear envelopes beginning to disappear. We have our sister chromatids. The unique thing about prophase 1 is that these homologous chromosomes are going to join together. Okay, and they're going to join together through chiasmata, which is the plural of chiasma. Chiasmata are the points, the actual physical touching points by which homologous chromosomes connect to each other. And when they connect, they're able to undergo a process called crossing over or recombination. And we'll talk about that in a few frames over. Metaphase 1 is very much so like metaphase and mitosis. These pairs of chromosomes, homologous chromosomes, are stuck to each other still through synapses, and they're going to line up on the metaphase plate. Okay, and they're still being moved by these microtubules that bind it to the kinetochores. This is just like mitosis. It's just that now we're dealing with homologous chromosome pairs. Okay, Anaphase 1, these homologous chromosomes, which have already recombined, you see a little red here on the big blue, zooming in up for you. See how there's been some exchange of DNA information? That's that recombination, that crossing over. They're going to be pulled to separate poles in anaphase 1. And in animal cells, this is going to create a cleavage furrow where we go through telophase 1 and cytokinesis. We have started with a diploid cell. Okay? We've had what? 1, 2, 3, 4, 5, 6 chromosomes in our parent cell. How many do we have in our two daughter cells? 1, 2, 3. 1, 2, 3. Okay, we have made 2 haploid cells. This is at the end of meiosis 1. You need to know when the cell is haploid, when is the cell diploid, how many do you make at each step? What are the chromosomes doing? What is the nuclear envelope doing? What are the kinetochores doing? What are the non-kinetochore microtubules doing? What is the job of non-kinetochore microtubules? These are all good questions that you need to know so we can understand this process. If we zoom in here, we see that the process that occurs in prophase 1 is called crossing over, or as I pull it up, recombination. Okay? These 2 homologous chromosomes are going to touch. This is actually an electron microscope image, or electron micrograph, from transmission electron microscopy, or TEM. This is actually a picture using electrons to see the presence of a homologous chromosome pair that has bound, that is binding in 2 different spots. And these spots are known as chiasmata. That's the plural of chiasma, which is different from the centromere, shown here. Anaphase 1, pull apart. Anaphase 2, we pull apart again. And we get 4 different daughter cells at the end of meiosis 2. But right now I just want to pull to the point of this crossing over that takes place during prophase 1. That's when recombination occurs. That's when you have the synapses forming. That's when you have the chiasmata touching. And that's when you have recombination. Recombination gives diversity. We'll talk about that in a few minutes. After meiosis 1 is complete, the 2 haploid daughter cells are going to divide again. There will be no more crossing over. Okay, we have no more synapses, we have no more chiasmata touching. We're just going to divide, and this happens just like mitosis. Okay, it's just another round of cell division. You can see here it lines up again in metaphase, pulls apart in anaphase. What's the protein that pulls apart these chromatids from each other? We know they're joined together by cohesin. What's the enzyme that actually pulls them apart? You need to know that. Okay, I'm not going to answer that question right now. I want you to look it up. But that's something that you definitely need to know. It's in the reading. The enzyme that pulls apart those chromatids in both mitosis during anaphase as well as anaphase 2 of meiosis 2. These sister chromatids separate. And when they separate, we get more cleavage furrow that begins, and now we have 4 unique daughter cells. That is meiosis 2. That is the end of meiosis. So what's the major goal? Well, we produced gametes. We went from diploid to haploid. In other words, we went from having 2 sets of chromosomes to only having 1 set of chromosomes. In humans, this would be going from 46 to 23. Okay? Also, we've created genetic variation through the process of recombination / crossing over. We've increased the number of types of gametes And in turn, we're going to increase the number of zygotes that can be produced. Whatever your N number is, depending on the species, so for humans it would be 23, you're going to raise 2 to that number. So for humans it would be 2 raised to the 23rd, or to the 23rd. That will give you the number of different types of potentially different gametes one person can make. Okay? And when that person mates with someone else, you're just going to multiply those numbers together, and that gives you the total number of possible zygotes that those two people could make. How unique are you that you are 1 in 2 to the 46? It's amazing. And not only that, but you have the recombination, which has put some of your father's genetic information and mixed it with some of your mother's genetic information on the same sister chromatid. And so you really are just increasing the number of genetic variation by going through recombination, thousands fold. This allows for the diversity that we see in life, why people have different shades of colors, different eyes, different hair types, different lengths, different heights, different body types, etc. This different behaviors. This is why we can have a world full of all unique people, because we have 23 chromosomes, we can have lots of different types of gametes, and we have recombination that can occur wherever those chiasmata are. Okay? This produces the diversity. Now, we've been focusing on meiosis, comparing it and contrasting it to mitosis, and that's something that I want you to do, is to go through and look at how different is mitosis versus meiosis. The cell types that are involved, the locations where it happens, the chromosomes that are involved, the types of chromosomes that are involved, the enzymes that take a role. Some of this will be the same, some of it will be different. Fill out a chart and come up with, compare and contrast those things. Now, meiosis focuses on the production of gametes which are housed in the gonads. And this leads us to the second part of the talk, which relates to 12.3, section 12.3 in your textbook, which is about cell growth and the control of cell growth. One of the most deadly cancers for women happens in her gonads, in her reproductive system. Ovarian cancer. Here, the ovary here, and what we see is actually the overgrowth of ovarian cells. And some of them are thought to actually be just epithelial cells, but there's some evidence that cells from your fallopian tube in women can also become oncogenic or cancerous. It's the second most common cancer in women, and it's highly deadly because the symptoms mimic other problems. You may have bloating or just abdominal pain and you don't know where it's coming from. And usually when it's detected only after the metastasis, which is just the movement of these tumor cells, these cancer cells, through the lymph nodes. You see here through the lymphatic system shown in green here. It can go and travel to the liver, to the heart, to the lungs, etc. It can be very fatal. And so if we want to understand this deadly cancer of your reproductive system and other deadly cancers, we really need to understand how cell division is controlled. Okay, if you're getting tired right now, feel free to pause this video, shake your head a little bit, do some jumping jacks, come back to it because we're about to talk about the molecular control of cell division and how it relates to cancer. Cancer arises basically from a breakdown in the control of cell division. Somatic cell division is tightly regulated. It has to be. Otherwise, cells will divide when they shouldn't. Cells will divide when there's little nutrients available, and they should not, because then they're going to just pull more nutrients and all the cells will die. Cells are able to respond to the extracellular environment to stop or enhance cell division. However, when mutations arise, and they can be environmental factors, it can be genetic predisposition, it could be certain behaviors that we partake in. Some of these mutations can be quite deadly because they can lead to a decrease in dysregulation of cell division, allowing for a cell to become oncogenic or cancerous. And sometimes it can even allow for the turning on of what we call oncogenes, which are just genes that actually promote the development of cancer. This comes from figure 12.15. Remember, this is the last part of chapter 12 in your book, talking about checkpoints of the cell cycle. Remember, we have these different phases. Okay, G1, S, and G2, they all make up of what? Interphase, correct? And then M is just for mitosis. Well, these different phases, we have checkpoints, which means the cell is going to stop and kind of assess itself, if you will, to see if it can go on to the next phase. Why do you think we need a checkpoint here between G1 and S? Think about it for 1 second, please. Well, what's happening in S? DNA replication. Well, I want to make sure that I have all the enzymes and all the material needed to efficiently replicate my DNA. If I do not, I could cause mutations to arise. I could call for failed DNA replication, which is one signal to cause my cell to die. Cells are not trying to die, but they will induce apoptosis, programmed cell death, if something is awry in their DNA, if there's been some damage to it. That's one signal for apoptosis. Where's the next major checkpoint? Well, it's right before M, right before mitosis begins. Well, I want to make sure I've doubled everything in G2. I want to make sure I have 2 pairs of the centrosomes. I want to make sure that my microtubules are intact. I want to make sure I've doubled my organelles. I want to make sure that I have everything so that at the end of mitosis, I will have 2 functional cells. And here we have another checkpoint at the end of mitosis, just to make sure that everything went correctly, that we did not put different numbers of chromosomes, that we have the equal amount of chromosomes in our daughter cells, that they are diploid for mitosis, okay? That they are haploid for meiosis. And there are actually molecular signals or mechanisms behind these checkpoints. One is CDK, which stands for cyclin-dependent kinase. Cyclin-dependent kinase is a kinase, that means it's going to phosphorylate something, correct? Well, this kinase, CDK, is not active unless the protein cyclin joins with it. And when cyclin here in purple joins with CDK here in orange, this complex is known as MPF, or maturation promoting factor. Okay? This MPF is going to allow for the cell to enter mitosis. If there is no MPF formed, you will not enter mitosis correctly. This is an example that special signaling molecules control the entrance and exit of major cell phases. See what happens here at the end of mitosis. The cyclin is degraded. Okay? We still have CDK hanging around, but we break down, we inactivate the cyclin. And as it breaks down, MPF is no longer present. This complex has been torn down. And this is the clue that we are now ready to exit mitosis and go into the G1 phase, so we can start the cell cycle all over again. If this process of checkpoints does not happen correctly, you can develop cancer. One can develop cancer. Cancer cells are dangerous because they do not exhibit the normal stop mechanisms of normal cells. Normal cells must grow on a substrate. This is called anchorage dependence. The cells in your body are not just floating around for the most part. They're actually attached to a matrix. Okay? They're attached to collagen, to some portion of the extracellular matrix that holds them in place. Okay? Cancer cells don't need anchorage. There are some cancer cells you can grow in lab that can grow in suspension, and they will not attach to anything, and they'll keep dividing and growing just fine. That's dangerous because that means that during metastasis, when these cells are floating just through the lymph system, they're still able to divide. And when they get to other different parts of your body, there's just more of them. Okay? Because they're just in the solution, just moving. Versus normal cells would not divide if they weren't adhered and stuck to something. Also, normal cells exhibit a process called density-dependent inhibition. See here in the picture here, these cells have divided and they're crowded the whole flask here. Well, if these were normal cells, they wouldn't grow anymore, for the most part. There are certain molecules on the membranes of cells that tells other cells, I'm close, we're touching, let's stop dividing now. Okay? This example here is that we're trying to pick up some cells, and now we have more cells that can grow in this space, because now we say, oh, there's no cells here, now I can divide again and make some more to fill this space. Once I fill this space, I don't need to grow anymore. Cancer cells don't do this. You can see here, not only do they keep dividing, they just start growing on top of each other. And I can show you pictures over in my lab when the cancer cells that I work with do this. They just grow on top of each other, and they will just keep growing until they just suck up all the food, all the nutrients in the media that we grow them in the lab with. Well, that's what they do in your body. They just keep growing and growing and growing on top of each other. They don't have this density-dependent inhibition like normal cells, which is why it's so dangerous. Cancer cells are dangerous because they don't respond to the normal cues that would keep them from dividing. They don't listen to the stop signs. They don't listen to the checkpoint. They just run right on through. They just keep ravaging without any anchor, without listening to their other cells nearby. They just keep dividing. They don't respond to the stop sign. And this is where you come in. This is where tying all this that we talked about together. Cell division of gametes, cell division of somatic cells, and what happens when cell division goes wrong. Our community, African American women, we're here at Tuskegee University, a historically black university, is particularly affected by cancer. One alarming statistic shown in this sign here is that breast cancer mortality, the rate of patients dying from breast cancer, is highest among African American women. This is true for a multitude of reasons. Lack of access to health care, or not going to the doctor, being scared, different cultural presumptions. But also, too, black women sometimes have a very deadly form of triple negative breast cancer, which was shown in one of your application videos, which is also available online, and it's in your module. We need you, students, Biology 230. We need your innovative ideas. How can we make cancer cells respond to the normal stop signs? How can we make them anchorage dependent? How can we help them stop dividing when they get too close to other cells? We need these ideas coupled with your dedication, because that's the only way to translate an idea to something that is tactile, that's something that something can help someone. We need your application of science. I'm trying to convey that we don't want you just to learn this to learn it, but we want you to learn it to use it, to help, to treat, to cure, to motivate, to educate. Okay? We want you to go and replicate the education that you learned here so that others in your community know better, can have better treatment outcomes. Simply put, the beginning of cancer is simply mitosis out of control. So if you understand how mitosis works, if you understand how cell regulation works, then we can start putting an N, big X, onto cancer. If the cell goes out of control and divides, this can lead to a tumor that could either be benign, which means that it has very little risk and will not metastasize, or it could be malignant, which is the scarier tumor because it could metastasize and evade the primary tumor site and go and circulate in the lymphatic system and the circulatory system and go and grow in vital organs such as your heart and your lungs and your liver and your stomach, etc., and be fatal. I've invested some time making this video. You've invested some time listening and learning from this video. You've invested time doing virtual labs, activities, reading, studying. Your parents have made an investment first by giving you their genes. Let's just start there. But you're more than just a copy, remember? You represent recombination, so you represent something new, some new hope that society can hopefully learn from. You represent a unique individual with the potential to lead to better therapies for cancer and other diseases that interest you. So we, including myself, need you, the fused product of meiosis, that's fertilization, right? Two gametes coming together, recombining, and producing you, a unique you. We need you to come to advance yourself, to challenge yourself, to learn in different ways that you may have not learned before, to study in different ways that you have not studied before, so that you can advance not only yourself, but your community, your region, and our world. This has been Dr. Griffin speaking on mitosis, focusing on meiosis, and the regulation and dysregulation of cells. For Biology 230, thank you. Have a great day.